It seems that everyone just suddenly got quiet, so we might as well start. There are a few things before spring break that I want you to give thought to. Number one is that our second exam is going to be the week after spring break on Tuesday, so the 24th is the date to remember. I will do review sessions on that day, both at 2:30 and at 7:30. The 7:30 one, of course, as always, is online on conferences to on Wild courses. In terms of the review, a couple of you had asked me if I could please get that done before spring break. I did it this morning, and it is posted, so you should be able to find, and maybe some of you already hunted it down on the announcement that I made this morning. It's posted as an announcement. Don't get overwhelmed by the length of that review. Bear in mind that it's all material that we have already covered. So it's nothing new. It's just another way to rehash the notes in a more boring way, right? The way we've covered it in lecture is by far the more riveting way to study and cover it. But it's just something that is there for your use should you think, wow, I didn't take very good notes on this section. I'm going to go ahead and check the review and see if there's something I should add to my notes for this section. Of course, also, if that happened and you dazed off in lecture, what is another good way to make sure you got it? Yeah, go back to the vodcasts. I mean, they really are one of the best ways to study, is to think, wow, I'm gonna recheck what happened in lecture that day, what did we cover, and go back to those. So see the review as just an added tool, but not something that's new or information that we haven't already been exposed to or that we haven't already covered. Couple of questions have come up about the poster. So we're changing things up a little bit this semester, and that is that I'm allowing you to have the poster for both Exam 2 and Exam 3. So you're going to get to use it throughout. And the more that we get jazzed about posters, the more that I realize that it is really, truly your medium for putting together all of the sort of internal connections onto something that I hope externally will help you. So the making of the poster, the synthesis, is really what it's all about. And students who do a super fantastic job on their poster get real mad at me after the exam because they're like, Rachel, I didn't use my poster a single time because I know this stuff so well from having rewritten and remaking and really putting together a poster that I know, right? And so it ends up being the process less than the product that is the most value to you. So I want you to put a lot of emphasis on the process and on writing and rewriting posters so that you get something that is really meaningful to you in the making. So with that, there are a couple of rules. And Macy was asking me this morning about, what are the rules on the poster? Can I put anything on there that I want to put on there? And absolutely, you can put anything on there that you want to put on there. I think Cassie was asking me about that. You bet. But here are the rules. It has to be on a single sheet. Now, the reason for that is not how some professors allow a note card, and the reason they allow a note card is because it's so small and you can't fit very much on it. The reason that I say a single sheet has nothing to do with how much you can cram onto it. Because if you want, you can use your shower curtain, and that's huge, right? You could cram absolutely everything we covered on it. So I fully expect that if you want to, you can cram everything it wouldn't even be cramming. The reason for a full sheet is that I want you to synthesize connections, right? I don't want it to be disparate, unrelated concepts that have no connection in your mind. I want you to think about how all of this goes together in a cell, and I want it to all show the connections within that cell. And or not show them, right? Because does the Ettner-Duderov pathway happen in the same cell as the Ebgen-Meyerhoff pathway? No. So you would want to draw a separate cell for the Ettner-Duderov pathway, or do something like Stan DeVore did in the poster I was looking at with some of you yesterday, where he made a fictional megakaryocyte, which was a cell with extraordinary numbers of metabolic pathways, and he clarified that. He said this is an unusual cell that happens to run both of these pathways. So it makes you think about how pathways are connected, how events within a cell intertwine, and you could put everything on there. So you could make your cell a yeast cell, right? You could put every darn transport system across the cytoplasmic membrane that you could think of, or across the mitochondrial inner membrane. You could add every dang thing that we have covered to date onto that cell, onto that poster, but it has to be of your own making, okay? No photocopies from books, because if it's that, all that is just blind memorization again. It's just bulimic learning again. But if you make it, then you're really synthesizing it, right? You're really thinking about how it goes together. So it needs to be of your own making. It also cannot have large blocks of texts or tables, because do tables occur in cells? No, right? And so that's the reason for those rules, not because I care if you have everything we've learned on that one poster. You can have absolutely everything, but I want it to represent a cell. I want it to look like a cell. Cells don't have tables in them. They don't have large paragraphs of text in them, notes, rewritten in them. They have, fluctuating, ebbing, and flowing, beautiful pathways, and that's what I want your poster to look like. Does that make sense? Okay, so once you know why those rules are there, I think you'll be able to put together a beautiful poster that can be an incredible resource to you, but it's primarily a resource in the process of making it, even more so than it is in the product that you end up with. Is it only supposed to be one-sided too? I don't care. It can be both. You know what might be really cool, right, is to have a yeast cell on one side of your poster with the things that are pertinent to a eukaryote, and then to maybe have a soil microorganism on the back that might be, say, an agrobacterium cell or an azotobacter cell that would run the Anaerobic pathway, and then would maybe have some of the characteristics we talked about in Norbert, right, that are bacterial characteristics. And so I think you can really, I'm putting that challenge out there, and I'd love to see, and so many of you ran up to me yesterday and said, Rachel, look at my draft of my poster, and it was rad, and I loved it, and those drafts are coming together. Keep bringing those. If you'd like to bring another draft tomorrow and show me what you have coming together, please definitely do so. So spring break might be kind of fun, laying out. One of the students told me yesterday that a good way to go is to get a refrigerator box that you can lay your poster on, and then you can use permanent pens on it, and it won't go through to your floor. So whatever works for you, be creative, and make it the poster that works well for you, okay? Good? 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 Okay, wonderful, wonderful. If I would stop talking, I think we would cover the rest of the material that's actually on the exam. So let's try to go there now. Do be aware that I will be working on the fever discussions over spring break. And also, I have gotten feedback to everyone but one group. One group's hypothesis came in pretty late, so I've still got to get you feedback. But otherwise, I feel like everyone has gotten feedback from me as of this morning. If your group wasn't that later group and you haven't gotten feedback, make sure that you do get a hypothesis to me that maybe it somehow got lost. But I'm waiting on revisions from most of you, and I'm totally flexible on when those revisions come in. My thought is this: how many of you are totally turning it around and running to Moab for spring break? Friday night, Moab bound, right? There's got to be some of you. Come on, guys, plan something fun for spring break, really. Don't let me down. Go do something fun for spring break. But my thought is that you definitely shouldn't let your hypothesis revisions sit over spring break. I think you're going to be real sad if they sit and get old and you're like, wait, what was Rachel talking about? Wait, what was I talking about? I don't remember any of this. If you come back after spring break, that's what's going to be what happens. So try to get those revisions to me if you can. Obviously, for that group that came in later, it may be harder for you to turn around those revisions when I finally get them to you. So, it's all dependent upon where your particular group is in the process. Okay, any questions, thoughts, concerns, marvels of the world? Okay. My poster's kind of lame. It doesn't have all the details on it. That's largely because it's hard to write with this pen, and I can't write all the structures. But at least it does give us an idea of what we were talking about last time and how we've seen all these magnificent pathways come together. Today, I would like to talk a little bit about the fact that not all sugars are glucose, right? We know that glucose is definitely the most quintessential, iconic source of energy. But there are other sugars as well, other monosaccharides. For example, galactose is another one that can enter into the pathways here. And I'm not showing the details. I'm just telling you that galactose is another monosaccharide. It can get converted to glucose 6-phosphate. Of course, we have the monosaccharides, fructose and mannose. And both of those can enter in at this point in catabolism. So there are many other sugar energy sources besides just glucose. And of course, even in our donuts, there are a few complex carbohydrates, maybe not a lot, but there are some starches. So today I want to talk about the fact that other fuel sources can enter into these processes. We did that a little bit on our map when we first entered into metabolism. But now let's see the details of how a starch might enter, how a fat might enter, and finally how a protein might enter into these processes of catabolism. So other monosaccharides such as mannose, fructose, and galactose, and others can enter into the glycolytic pathway. Starches, disaccharides, things like lactose, sucrose can enter into catabolism as well. They can be converted into their monosaccharides, which can get added into Ebden-Meyerhof pathway. Starches, we know, are the longer polysaccharides. This particular polysaccharide shows a little piece of an amylopectin molecule, or maybe glycogen. It's a branched polysaccharide. Now, we know that bacterial cells don't particularly do much in the way of phagocytosis, right? They're not going to reach out and get the big gulp of very huge macromolecules. And so instead, they have to be tricky. They have to secrete enzymes that go out into the extracellular space and clip down these giant polysaccharides into manageable pieces. And so they secrete enzymes that will either clip the 1,4 linkage or the 1,6 linkage in this polysaccharide. Wait, why do we call it 1,4 and 1,6? Good. Good. The carbon that it's linking. It's this bond, this glycosidic bond is linking carbon 1 and 4. This one is linking carbon 6 and carbon 1. So that's where they get their names. So those are the glycosidic linkages that a bacterial cell is going to have to secrete enzymes that will degrade those bonds, that will break down those bonds. And it does. It secretes two enzymes, oligo-1,6-glucosidase and alpha amylase. Alpha amylase cleaving the 1,4 glycosidic linkage, oligo-1,6-glucosidase appropriately cleaving the 1,6 glycosidic linkage. Now, as I said, these are secretory enzymes. And remember how sometimes looking for metabolic capacity is the key to identifying unknown bacteria in the lab. Remember how we talked about looking for cytochrome C oxidase as a quintessential identifier of things like Pseudomonas. We can also look for the abilities to secrete oligo-1,6-glucosidase and alpha amylase as a key to identifying certain bacteria. For example, who had Bacillus subtilis, the endospore former for your unknown the first time round, right? If we were to put that unknown organism onto a plate with starch in it, and we were to drop iodine onto the starchy plate surrounding the bacterial growth, the iodine would bind to the starch. But if the starch had been depleted because the organism had secreted these two enzymes, suddenly the starch would be gone surrounding the bacterial growth, and there would be no starch for the iodine to bind to. So in this test, which we call the starch hydrolysis test, which organism, the one on the left or the one on the right, produces oligo-1,6-glucosidase and alpha amylase? The one on the left, right? Because you're noticing there that the halo around the bacterial growth indicates that the starches have been depleted in the medium surrounding that bacterium, whereas here, this organism has failed to deplete those starches. It hasn't secreted those enzymes. So again, this capacity can be a means by which we identify organisms, whether they can degrade those polysaccharides or not. So cellulose is another polysaccharide that some bacteria can degrade. Cellulose, of course, also is a polymer of glucose residues linked by a slightly different kind of glycosidic bond, a beta rather than an alpha bond. And so some bacteria secrete the enzymes called cellulases that degrade that particular polysaccharide. There are some of these that are very, very famous because they are found in the rumen. For example, in the rumen of cattle. And in that environment, it's like a huge fermentation vat where the cellulose that a cow eats is then looked at as an energy source by many of these bacteria. They degrade it first into cellulose, which is the disaccharide that repeats in cellulose. And then finally, they degrade that disaccharide into acids, such as propionic and butyric and acetic. So remember, that's a kind of mixed acid fermentation that we talked about being a common fermentation in certain bacteria. And then the cattle look at those byproducts and they're like, woohoo, dinner. So, right, suddenly those symbiotic bacteria have created fermentation byproducts that are a fuel source to the cattle themselves. Interestingly enough, guess what is also a fuel to the cattle themselves? The bacteria. Excellent, Nathan. Yeah, so they actually literally are digesting some of the bacteria, which are their symbionts, as a nutrient source as well. So this is pretty cool action that's going on within the rumen of all of those ruminants, not just cattle. It's kind of cool, and I just have to shoot this in here. There's something on the order of like 10 billion microorganisms per gram of wet weight in the rumen. So like, yeah, I know, it's just nuts, the microbial density, right, that we see within that ovosome. It's really amazing. So the glucose subunits released when polysaccharides are digested can enter into the glycolytic pathway in these bacteria. First, they degrade it to disaccharides, then to monosaccharides, and then finally they enter into the Ebden-Meyerhof or what have you fermentative pathways that they are running. So not only can bacteria degrade these polymers, right? They can degrade the starches and the cellulose, but they also can often degrade compounds that most organisms would look on as being completely inedible. So we've talked a lot about this, and even did yesterday in lab, the fact that many bacteria see things like TNT or the alkanes oil, right, as being a fuel source. We call those compounds recalcitrant. Recalcitrant is just a sexy-minded term for compounds that are difficult to degrade. They're resistant to degradation. But many bacteria see those recalcitrant compounds as energy sources. So when we think about, wow, those have to enter into catabolism in different ways, you can see how the complexity of the metabolism of Alcanivorox, for example, which is an alkane-degrading bacterium, may have to adapt to its food source, which is oil, in order to be able to live on that food source. Now, many of them have some of the same pathways, right? And in a moment, we're going to look at how fats are degraded, and I bet you can think how that might transfer over to how alkanes or oils would be degraded as well, right? Very similar. So lipids, fats. And of course, in our donuts, this is also one of the energy sources that we are taking in. And briefly on our map, our big picture map of metabolism, we said, hey, you know, try acylglycerols. And a couple of you came up to me after the first exam and said, what, am I supposed to know what a TAG is? You know, TAG, triacylglycerols, fats. The old school name, triglycerides. So those are some of the names that a fat goes by. Lipids, right? It is one type of, in the macromolecule category, lipids. So what kind of enzyme would a bacterium make if it degraded lipids? Lipases, yeah. And lipases cleave ester bonds. They cleave these ester linkages right here. So just clipping these down. And what that leaves is a 3-carbon glycerol molecule and 3 long acyl groups, right? Fatty acids, basically. So fatty acids are kind of crazy. They are not particularly life-giving to a cell. They actually act as detergents. So most cells don't like to keep fatty acids around for any extent of time, but they will just go ahead and degrade them, like eat them as a food source. So let's think about what might happen to a glycerol backbone after this degradation. So first, lipases cleave, so that now we have the smaller subunit. And of course, a big part of that is to have something small enough that a bacterium can bring it in, right? Size really does matter. It's got to be something small enough that the bacterium can get the subunits in. Yes, it was a joke. So these side chains will come in separately from the glycerol backbone. Who can think about Embden-Meyerhoff pathway, right, glycolysis? Where might a 3-carbon molecule enter into that catabolic pathway? At what point in the Say it again? Yeah, good. Right as soon as we start to make, to have the triose or 3-carbon stage, absolutely. Anybody remember what those molecules were, the first of the trios? Glyceraldehyde, 3-phosphate, and dihydroxyacetone phosphate. Excellent job. Good. You guys are rocking it already. So we would see it enter in as one of those 2 molecules. Why might I say, yeah, doesn't really matter which one we think of it entering as? Because they're in constant equilibrium with one another. I love Trey's body language. They're in constant equilibrium with one another. So they'll enter as glyceraldehyde 3-phosphate if we're talking about further catabolism. But it's interesting to note that with respect to what a cell is typically doing when it's living off of fats, it's usually not going to degrade that glycerol backbone. And here's why. If a cell is living off of fat only, it's usually wanting to make some sugar. So it's typically going to use the glycerol backbone as an anabolic precursor metabolite, a LEGO, to build glucose. So that's why for all of our future hypothetical calculations, and you're about to do one, we won't add in the energy that would come from the glycerol backbone, because we're going to assume that most likely its fate would be as a precursor metabolite, as a LEGO for biosynthesis. So now let's think about the energy that could come from the fatty acids themselves. So these long hydrocarbon chains, the fatty acids. So the glycerol backbone, that will enter in glyceraldehyde 3-phosphate and intermediating glycolysis. Now, here is the kicker. The fatty acids, the long hydrocarbon side chains, will enter into catabolism 2 carbons at a time. That is, they will be broken down in 2-carbon units. So this will be one breakdown product, right? This will be the next. So 2-carbon units at a time. Where in catabolism do we first see a 2-carbon unit? Where did we first see a 2-carbon molecule arriving? Yeah, excellent. So at the end of the transition step, acetyl-CoA is a 2-carbon moiety. That is how the 2-carbon units of a fat will enter in. They will enter in as acetyl-CoA. AKA, said another way, they enter at the top of the TCA cycle. Okay? So that being said, and we will write it down, I want you to count and tell me how many acetyl groups will result from the degradation of one of the fatty acids. This carbon counts. Okay, good. 7, right. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 carbons. They enter 2 carbons at a time, so 7 acetyl groups. So now I want you to work with someone next to you, pull out a scrap piece of paper, put your first and last names onto it, And I want you to tell me, okay, now listen up. I want you to tell me how many protons could be pumped by an aerobically respiring chemoorganoheterotroph if it degraded all 3 of the fatty acids in this fat. Okay? So go to work at that. I'm going to give you like about 4 to 5 minutes. And you guys are going to pass those in. And I'm going to give you some extra credit, and then we're going to discuss them later. So, okay, I'm going to repeat it one more time. How many protons could be pumped from the catabolism of all 3 of those fatty acid side chains? Oh yeah, pictures help, right? Yeah. Pictures help, calculators help, sure. Be careful with that table, right? Because it's an acetyl group, not a glucose. Right? So just make sure you're aware of how many times the TCA turns. Yeah. So as you're starting to work, let's repeat this one more time. How many protons could be pumped by the proton pumping members of the ETC, right? How many protons could be pumped if all 3 of those side chains were degraded? So what's one of the first things that you need to determine? How many times what turns? TCA. Yep. A good place to start is how many times in total does that TCA turn? And then what are the yields from one turn, right? Maybe it helps to have pictures. Good. Okay, good. So 7 acetyl-CoA for one side chain, right? But how many in total? Times 3. So, good. So now think about the yield from the TCA for 21 turns. So did you figure out how many times TCA would need to turn?
If we have
21, good. 21 times, right. So then think about what is the yield per one turn. Okay. Good. We're starting to get to the point of knowing that the cycle turns and X number of times. And then think about the yield per turn. And then what are you going to need in order to get from there to protons? Yes, a conversion factor, right? However many protons per good. Good. 21 acetyl. Good. Good. Okay, that works all right. But make sure you're aware of how many, okay, you're going to come up with a slightly different answer that way. Try to be aware of how many NADHs are made, how many FADH2s are made. Yeah.
Isn't there 3 NADH per acetyl-CoA?
Yes.
Okay, so then you would do that times 10 hydrogens
Good.
per NADH?
Good. You're well on your way. Yes, you're well on your way. Potentially. I can't remember exactly what number 20 on the homework said.
It's like the TCA cycle turns 8 times.
Right.
How many protons?
Right. Yeah. Yep. And Zach is raising a good point. He said, hey, this is kind of like number 20 on the homework. Yeah. Whoop, whoop. Oh, yeah. Got it? I can see the right answer on this sheet. So bring them up. Bring them up. You guys just pile them right here. Get a little bit of action. Dance a little on your way up to the stage. So did anybody, when you finally saw that number of protons pumped, did anybody think, wow? Did anybody think that? Wow. Why did you think that, John? Wow, that's a lot of protons. And maybe it got you thinking, wow, fats are packed with power, right? Fats are actually the most efficient fuel source. Per carbon, they yield the most ATP. So that's an interesting thing to ponder and to think about. I want to talk briefly, though very briefly, about how proteins can enter catabolism. Right? We know that fats are an efficient fuel. Proteins, they are not so much of a good fuel. We know that a big reason for that is that they have to be, each and every amino acid that enters catabolism has to be deaminated. And that deamination generates what? Ammonia. And ammonia is toxic, and ammonia has to be secreted. I got a great email from Sammy while I was gone. She was like, what about nucleic acids? Like when you eat DNA and RNA, aren't you also having to deaminate those? You are, right? So we could say as a fuel source, they also have to be detoxified. But let's look at where an amino acid might enter into catabolism, and they enter in different places. Once a protease degrades the peptide bond between 2 amino acids within a protein, we know that liberates an amino acid. Does anybody hopefully recognize this particular amino acid that was liberated here? So we're saying that the little blue ball is this amino acid. What is that amino acid? Glutamate. Yeah, good. So that is the acidic amino acid glutamate. And just by saying that name, you might think to yourself, glutamate. I wonder if that looks like alpha-ketoglutarate, an intermediate in TCA. And in fact, it looks almost exactly like alpha-ketoglutarate once it is deaminated. And it enters pretty darn directly into catabolism as alpha-ketoglutarate. So this is a good example of how the backbone can enter into normal metabolic pathways, where we can see an amino acid getting catabolized. So even though we know that proteins are not the preferred fuel source for a cell, they can definitely enter into catabolism. There are other nuances to that, but I'm going to leave it at that point. And I'm going to say that this is the last of the exotic fuel sources that we're going to look at, and we're going to kind of end that particular coverage there. Does anybody have questions on protein catabolism or fat catabolism? It's worthy to say that amino acids, like say, for example, alanine. Alanine can enter in as pyruvate. Other amino acids enter in as other intermediates in the TCA cycle. So it's not just, this is just one example of one place that a protein, that an amino acid might enter in. Yeah, Eric.
Is there a special enzyme you need to rearrange each amino acid to be able to TCA cycle?
Yeah, his question is an excellent one. So what he's asking is, are there special enzymes that are needed in order to incorporate these amino acids into as intermediates in catabolism? Yeah, so there's actually a set of enzymes called transaminases, and they actually take the amino group from the amino of the amino acid and transfer it generally onto another amino acid, which then transfers it onto the first. The enzyme is the first pathway in the urea cycle, then transfers that, right, and eventually there's a long urea cycle that I'd love to sit and talk to you about if you'd like, that eventually gets that transferred onto or makes urea from it. Yeah. Yeah, it's a great question. Sammy's question is, do most amino acids enter into the TCA cycle? A lot of them do, but not all of them. So like alanine is going to enter as pyruvate, which is further upstream. Aromatic amino acids sometimes enter into like pentose phosphate pathway. So there's a lot of varying places, but a lot of them do enter at the TCA at varying intermediates. And there's also varying numbers of stages that have to occur in between. The entry of glutamate is one of the simplest, and it's why I used it as an example. Yeah. Very great question. You guys, your minds, they're going. I love it. I know that you're ready to finish micro when you start asking questions where I go, I don't know, or I say, I'm not sure anyone knows. And that's what I hope your future is always like, right? Always asking questions and not believing answers and doing more research. So on that note, speaking of asking questions and doing more research, I think probably one of the most explosive areas of understanding of the prokaryotic world is in the concept of rock eaters. And I actually had a student a couple of semesters ago. Actually, I believe he's now a graduate student. He made this really awesome picture. And I know I was talking to you about the never-ending story a while back and the rock eater in that. So did anybody take me up on actually going and watching the never-ending story? Okay, that is a good spring break activity. It's your assignment over spring break. Is to watch the never-ending story. But in that, there is this fantastic character called the Rock Eater. And so this is the picture that Cameron made, chemolithotroph rock, right? And that is, by definition, what a lithotroph is an organism that degrades inorganic chemicals as its fuel source, aka it's a rock eater. So I want to talk about that because prokaryotes are the only form of life that is capable of eating rocks, of degrading inorganics, and of allowing electrons to be ripped and stripped off of things like, say, your household ammonia cleaner, right? Something see ammonia as their donut. Some organisms literally see hydrogen gas as their donut. I mean, thinking about the kinds of things that a prokaryotic organism can degrade as an energy source is just, it's mind-boggling. And I hope that some of you are thinking about the applications of some of this as well. So prokaryotes are alone in their ability to use inorganic compounds as their energy source. And I was talking with Jinsey and a couple others yesterday, right, about the meanings here. These collectively, if we put this all together, would be chemolithoautotrophs. So what is the autotroph? One more time, just as review. Autotroph means that these do what? Yeah, they make their own food. So chemolithoautotrophs are primary producers. They are making organic carbon themselves. They're using CO2 as the starting precursor metabolite for that. The litho means that they're degrading inorganics to get the energy by which they then fuel the processes of what we might think of as the dark reactions of photosynthesis. So it's kind of crazy to think, right, here's something that is not a photosynthetic organism. It gets its energy from inorganic chemicals, but it does the dark reactions of photosynthesis. That is, it creates sugar from carbon. So that's what the autotroph part of this means. So think that through a little bit. So many of these organic compounds are byproducts of anaerobic respiration. So remember yesterday, or Monday, how we talked about the fact that some bacteria will use something besides oxygen as their terminal electron acceptor. Remember we mentioned that things like nitrate, sulfate, CO2, right? All of these things that the anaerobically respiring bacteria said, okay, you can be my terminal electron acceptor. You're not as good as oxygen, right? Not as high of a standard reduction potential as oxygen, but you'll do, right? You're highly oxidized. I'll dump my electrons off on you. And when they dumped electrons off on these, they made things like hydrogen sulfide gas as a byproduct. Well, chemolithoautotrophs look at the byproducts of this and say, yum, donuts, right? So a chemolithoautotroph eats hydrogen sulfide, eats methane gas, eats nitrite, eats ammonia, right? It eats the byproducts of anaerobic respiration. Lovely kind of interrelationship, right, between those types of organisms. So thinking about the possible fuel, reduced fuel sources, hydrogen gas is the simplest. The hydrogen bacteria will rip and strip their high energy electrons off of their donuts, which is hydrogen gas. The sulfur bacteria rip and strip their high energy electrons off of things like hydrogen sulfide. So highly reduced forms of sulfur will be their energy source. Lots of high energy electrons to rip off of there. We see the iron bacteria ripping their electrons off of the ferrous form of iron, creating, of course, the ferric form. So they're oxidizing the iron. Some bacteria are so cool that they oxidize both iron and sulfur simultaneously. And I'm going to show you one of those in a minute. The nitrifying bacteria, two types. One of them chows down on nitrite, the reduced form, right? When nitrate is used as a terminal electron acceptor in anaerobic respiration, nitrite gets formed, and then chemolithoautotrophs say, yum, dinner, right? So nitrite serves as their source of electrons. What cycle do you think the nitrifying bacteria are particularly important in? The nitrogen cycle. They actually catalyze a whole corner of the nitrogen cycle. So clearly they are very important soil microorganisms. That's their environmental niche. So we can think of these as being not so different from us in this way. They only see donuts as different, right? The only thing that's different about their metabolism is that they rip and strip their high energy electrons off of things like hydrogen sulfide. But then everything's pretty much the same. They still send their electrons to an electron transport chain. They still use the energy from them to pump protons. Most of them still respire aerobically. That is, they use oxygen as their terminal electron acceptor. So they do a lot of things the same way we do, they just eat different stuff. But because they eat such different stuff, they clearly survive in unique niches, right? They survive in areas where other organisms would think it was toxic, because they do look at things that are often thought of as toxic byproducts for other organisms as their fuel source. So they thrive in extreme environments where there is a high concentration, for example, of heavy metals, right? Where they say like dinner, you know, and everything else is like, wow, heavy metals are toxic. So I want to tell you about a very special, well, a bacterium that's very special in my mind, called Bacillus Ferrooxidans. So thio means what? Sulfur. So thiobacillus is a sulfur user, right? It sees sulfur as a source of energy, so hydrogen sulfide. But then ferrooxidans means ferro, iron oxidizer, right? So this is a special bacterium that sees both iron and sulfide as an energy source. Iron sulfide. Wait a minute. Iron sulfide. What is that? When I was a little girl growing up in Leadville, Colorado, which this is just how much of a mining town Leadville is, we all had big chunks of fool's gold. And in fact, we had this huge chunk of fool's gold that served as our favorite doorstop. And it's fun, my mom even brought that when she finally, only a couple of years ago, made the move from Leadville to Laramie. She brought her big, huge fool's gold doorstop. And I still think about it so fondly. So Bacillus Ferrooxidans looks at fool's gold, which I should have brought it with me today, and it says donuts. And it chows down on the massive amount of fool's gold that's found in Leadville. But when it does that, when it eats fool's gold, here's what it does, right? It oxidizes the ferrous form of iron, and it oxidizes the sulfide form of sulfur, and it creates ferric hydroxide and sulfuric acid. You got it. So what a badass Bacillus Ferrooxidans, right? It is making one of the strongest acids out there. A bunch of sulfuric acid is getting released from this oxidation, and the sulfuric acid is doing two things. Number one, it's dipping the pH way down, and so that low pH is solubilizing a bunch of minerals in mine tailings. So these mine tailings end up taking on these appearances that I showed you some pictures of in lab, of this really, really orange color because of the production of sulfuric acid. But the other thing that it does is who's ever, and like I have the dubious honor of having done this more than once, who's ever spilled sulfuric acid in the lab? Yeah, what happens? Multiple bad things, but what's one? Remember? I spilled a lot of it on a bench surface. It's hot. It gets real hot, right? So it actually heats up. It will sometimes even get smoky. It's hot, right? And it's also extremely corrosive, right? So sulfuric acid will create a very low pH, very hot environment. And so in some mine tailings, you will actually see them smoking. Have you ever seen like a coal refuse pile smoking? And it's because of all the sulfuric acid. And it's like dipping in pH and sulfuric acid is going high. And then all of a sudden, other bacteria that are thermoacidophilic start to grow in these environments as well. And it creates a really unique environmental niche. Obviously, a very highly polluted one. Leadville has been an EPA Superfund site, which is a whole nother story if you want to hear about interesting like social dynamics between how the EPA relates to locals in a small mining town. It's pretty much a shit show. But if you want to talk about that sometime, I would love to do so. But on that note, let's just end for the day thinking about Bacillus Ferrooxidans and seeing fool's gold as donuts. You guys are running away. That ends exam coverage right there. So that's it for exam 2. Yeah.
I was reading an article about bacteria in oil. They were describing how they, separate those from other types of bacteria.
Sorry, one more time on the beginning. Bacteria that do what?
Eat oil.
Yeah.
And I was wondering, is there a way to select for bacteria that eat the oil faster than others?
Yeah, absolutely. Absolutely. And that is something that would be covered under the idea of bioaugmentation, is it's a little bit less invasive than actually just adding in outside cultures, right? Like they say ex situ versus in situ, like bioremediation. And if you're bringing in outside organisms that aren't there, it's risky in the sense of like changing the balance of the natural area. But on the flip side, bioaugmentation is pretty common to like select for the group of bacteria that seem to be already naturally degrading the oil the most rapidly. And that's what's done more commonly. And sometimes it includes just adding like things like fertilizers in order to increase the rate of growth of the population that's already degrading oil. And yes, it's possible, say you wanted to know, is there a variant of this one that's already degrading oil that is more rapid, like, and could we augment that one, you know, in higher concentration? Sure. I mean, you could most certainly, even in a lab environment, right, isolate one that is, you know, whether it be a mutant or, you know, something that has a higher rate of degradation. Yeah, yeah.
So would you, do you have to, like, I guess, make like lots of cultures and try to find one that?
Yeah. And that would be, so you could almost think about like some sort of colorimetric assay where the, you know, the oil had, you were able to look you know, through some kind of spec that would show you like, you know, okay, oil is, or even simpler than that, around a colony, like here's this colony is clearly degrading more rapidly because, you know, like even something as simple as like, you know, when we do the lipase assay and we look for fat hydrolysis, right, a larger area, and you could pick that one and say, you know, let's, yeah. It would be cool, Eric, to think about methods that you might use to do that, you know, even on a simple level. Yeah.
Well, I was just thinking about what you were saying about the coal, like the mining.
Yeah. Yep.
So there's the coal seam in Sheridan that supposedly caught fire a long time ago, and in the winter it steams.
Yep.
Is that really?
It most likely.
going on?
Yeah, most likely. The other thing that can happen is like methane production that can lead to like a spontaneous combustion, but it's the same idea, right? And you know, yeah, the steaming and sulfuric acid certainly can ignite if there's something flammable in the environment. Yeah.
Okay.
So I mean,
just says, oh, it's burning.
if it seeps out and it contacts something flammable, sure.
Okay.
Yeah. Yep.